Today we will be discussing Middle East Respiratory Syndrome, also known as MERS CoV.
What is MERS? MERS stands for Middle East Respiratory Syndrome. It is an illness caused by a coronavirus called MERS-CoV. It is zoonotic, meaning it is a virus that is transmitted between animals and humans. Our reported cases were linked to the Middle East, and it is contagious through close contact. Location of infections. It was first reported in Saudi Arabia in September 2012, but it was later identified that the first known cases of MERS occurred in Jordan in April 2012. Most people who got infected was through direct or indirect contact with camels. It is believed that it originated in bats and was transmitted to camels in the past. There was 2 confirmed cases of MERS in the United States, and it was due to a recent travel. This happened in May 2014, one in Florida and the other in Indiana. Both of them traveled to the U.S. from Saudi Arabia. They were both hospitalized in the U.S. and were discharged after a full recovery. Other outbreaks happened in the Republic of Korea in 2015, and it was associated with a traveler returning from Arabian Peninsula. This is the 2012 through 2015 global map of countries with confirmed cases of MERS. The darker the color gets, the more cases there is.
Here are some graphs that we found as well, just showing the spikes of infections in different areas. We have also a summary of the total amount of cases that have been reported and the amount of deaths, as well as how many countries that have been reported as well.
MERS comes from the family Coronaviridae. It is the first C lineage beta coronavirus associated with human infections. Its close relative is SARS, which stands for Severe Acute Respiratory Syndrome. Morphology. They are named for the crown-like spike glycoprotein on their surface, and this is where viral entry takes place, receptor binding, and membrane fusion. It has a membrane glycoprotein that's part of the triple membrane spanning. It has a small envelope glycoprotein. It is an envelope-positive sense single-stranded RNA encoding at least 10 open reading frames. The nucleocapsid phosphoprotein are essential for RNA binding, synthesis, and translation. The virulence factors are the type 1 interferon system that plays a major role in antiviral innate immunity. It is subdivided into the interferon induction pathway leading to transcription and secretion. MERS life cycle. First, MERS-CoV binds to the receptor DPP4 through spike protein. It enters the host cell by membrane fusion or endocytosis. It then releases the viral RNA into the cytoplasm. The RNA genome is then replicated and viral proteins are produced. Virions are then released from the infected cells through exocytosis.
Now we will be discussing the pathogenesis of MERS infections. The direct route of entry is primarily through the mouth and nose, very similar to COVID-19. Pretty much any close contact. Per the CDC, close contact means being around somebody or living with somebody that has a confirmed case. Direct contact with respiratory secretions or body fluids. Examples of those being hugging, sharing eating or drinking utensils, talking to somebody within 3 feet versus the 6-foot social distance that we're going through now, as well as direct touch with somebody else. Before the discovery of MERS, SARS was considered to be the most pathogenic coronavirus. The MERS mortality rate is about 30 to 40%. SARS was only about 10%. And this exact version of the virus does replicate in airway epithelial cells. There is a bit of a range for when these symptoms start to present. Typically, it is between 5 to 6 days after exposure, but it could be anywhere between 2 and 14 days after exposure. Most of the confirmed cases do have fever, cough, shortness of breath. Some also have vomiting or diarrhea. Many cases have more severe complications, such as pneumonia or kidney failure. And again, the mortality rate is much higher at about 30 to 40%. As you can see in these images, we have a before and after picture of a patient. We have chest x-rays. The first picture, the lungs are nice and clear. In the second picture, you can see much more of the opacities, as is typical with x-rays of pneumonia. There are 2 different types of tests to determine if somebody has a MERS infection. The first one being molecular testing, which looks for evidence of an active infection. This test is a real-time PCR assay, and this detects the viral DNA. The other test is a serology test. This one looks for evidence of a previous infection, so this test is looking for antibodies. One of the primary tests that they use is the ELISA test, which we learned about earlier in the semester. Just because of the limited amount of patients and limited data pool that we have for this particular strain of coronavirus, it is unknown if those that test positive but are not currently symptomatic are still spreading the infection. Now on to prevention and treatment. The basic prevention is very similar to what we're going through with COVID-19 right now. Basic hand hygiene, washing your hands, making sure you're covering your nose and mouth to prevent those respiratory droplets from getting into your system. Avoid touching your face in close personal contact. Disinfecting surfaces frequently and thoroughly. Treatment is pretty much supportive and symptomatic. There is no current vaccine for this virus, and there's no standard antiviral treatment. However, there have been some studies that have shown that some immunosuppressant drugs may inhibit the replication of this virus, but there is no proven treatment that this is effective.